Hi everyone, this is Physical Principles of Quantum Biology, Chapter 6, Dynamic Control of DNA Repair in Photolyase. This is the 6th chapter of the book, Physical Principles of Quantum Biology. I wrote in collaboration with my wife, Brandy Babcock. I'm Dr. Nathan S. Babcock, and as I mentioned, today we will be covering Chapter 6 on DNA repair by photolyase. So what is photolyase? First, let's consider the cryptochrome photolyase superfamily of proteins. These are enzymatic proteins that basically act as light-sensing receptors in the case of cryptochromes and photoactivated enzymes in the case of photolyases. They're very similar, but they're not exactly the same. So, for example, cryptochromes are light-sensing receptors. They're light receptors, similar to the light receptors in your eye. They govern circadian rhythms. They have disordered C-terminal tails, and typically they lack DNA repair activity, although some fungal cryptochromes were found to repair DNA in controlled circumstances in vitro. Now, photolyases, on the other hand, are light-activated enzymes. They actually carry out chemical transformations. They repair ultraviolet light damaged DNA. They do not have C-terminal tails, and they are present in bacteria, archaea, fungi, plants, vertebrates, but not specifically in placental mammals like humans. Now, cryptochromes and photolyases are in the same family, even superfamily of proteins, because they share similar architectural features. They're both ultimately light receptors. They carry out either signaling or DNA repair functions. They have homologous protein folds, molecular weights of 50 to 75 kilodalton, and a primary chromophore that's a Flavin type chromophore. That is a Flavin adenine dinucleotide molecule buried in the center of the protein. Typically, cryptochromes and photolyases also have an additional antenna molecule, such as a folate or a pterin, to broaden its light absorption. So if we take a closer look at this example of photolyase, we can see here it's docked to a strand of DNA. Looking a bit closer, we can see the Flavin adenine dinucleotide cofactor in the molecule. That's a prosthetic cofactor added to the protein strand after transcription. If we look a little further back, we can also see a hydroxydeazoxyriboflavin. That's an 8-HDF antenna molecule. That's the additional molecule that helps the flavin gather light. Now it's also surrounded by, as I've shown in purple, a variety of tryptophan residues. This amino acid carries a bicyclic indole ring. It's highly aromatic, and it also has light-sensitive properties. So the catalytic cycle of a photolyase carries out DNA repair, and it produces DNA repair yields near 80%, even in some cases approaching 90%, through synergistic optimization of multiple catalytic steps. So first of all, the enzyme has to bind the DNA. It absorbs a photon of blue light. This photon produces an exciton, what we call a photoexciton. For the non-physicists in the audience, an exciton is an electron-hole pair. It's an excited electron that leaves a hole in the electron sea, so to speak, when it becomes energetically excited by absorbing a photon. After the excitation occurs, there's forward electron transfer. The electron's transferred to the DNA, where that excited electron now breaks certain carbon-carbon bonds in the DNA. Now there's reverse electron as the electron comes back to the flavin, recombines. In some sense, the electron falls into the hole it left when it was excited, and the enzyme finally releases the DNA in the final step of the catalytic process, the enzyme can then go on to repair another damaged piece of DNA somewhere else. Now, before I move on, just one note for the structural biologists in the audience. The Photolyase I've shown here, this 3D rendering of it is drawn from protein databank entry 1TEZ or Z for the Canadians in the audience. DNA recognition and docking is the first step in the catalytic process. Molecular recognition of DNA by enzymes such as Photolyase involve non-covalent interactions. Now, these kind of interactions include dipole-dipole coupling, electrostatic interactions, induced dipole coupling, these kinds of classical interactions, but also non-classical, that is truly quantum mechanical London dispersion forces, which are particularly important in coordinating the double helix structure of the DNA strand. So you can imagine initially Photolyase binding to DNA. As it does so, this is in fact the first step in the recognition process, as a repair enzyme like Photolyase needs to find a DNA strand while floating around diffusing in three-dimensional space. Now, after it's initially docked to the DNA, the next step is for it to scan and ultimately find a damaged place, a damaged location on the DNA. So a repair enzyme will search for damage to the DNA, sliding along the DNA strand, essentially in one dimension, until it finds and docks with a damaged piece of the DNA strip. Now, if we look at this process, as the DNA repair enzyme slides along the strand, the search for damage is understood to take the form of a classical search model, which is a classical random walk. That means that, first of all, the protein, you can imagine sliding along the DNA strand in the one dimension defined by the DNA strand and occasionally hopping diffusively to another strand in 3D. When it binds to the DNA strand, the enzyme slides along backwards and forwards, either randomly backwards or forwards, moving along in what a physicist would call a random walk. Now, confining the enzyme to that one-dimensional pathway along the DNA strand where it can only go forward or back, it can continuously scan for any structural anomalies, and if the enzyme reaches a target DNA region, the high affinity basically allows it to clamp to the enzyme at the place of damage. So we have an induced fit model that says that the enzyme finds a place where the DNA is damaged. There's basically a little kink or changed structural properties at the damaged point, and the enzyme naturally clamps onto this position. Now, the very high efficiency of this search has prompted some to suggest that the classical random walk model could be enhanced by certain features of a quantum search. We call this facilitated diffusion. So repair enzymes were proposed to use DNA-mediated charge transfer to detect DNA damage. In this case, the search mechanism relies on the cooperation between enzymes bearing iron-sulfur clusters. And the repair model proposes that DNA-mediated charge transfer, you can imagine quantum mechanical wave-like transfer mediated by the London dispersion forces along the DNA strand, allow it to search for and ultimately locate damaged parts of the DNA much faster than pure classical 1D diffusion. That is a classical random walk. And certain species of enzymes, such as base excision repair enzymes, and of course DNA photolyase enzymes, the topic of this talk, in some species do contain these iron-sulfur clusters, which would enable this kind of model at least to be plausible. And so I've included a few citations on this page you can look into if you're interested in the possibility of quantum-enhanced search for the damaged piece of DNA. Now, with that in mind, there are 4 key stages of DNA repair. First of all, there is dark state photolyase DNA binding. We're not thinking of any photons at this stage. The enzyme, if it's floating around, it attaches to the DNA, it slides until it docks and finds the UV damaged part of the DNA strand. And this would be, for example, DNA with a cyclobutene pyrimidine dimer. I'll talk about that in a moment, but this is DNA with a CPD lesion. Next, having located the damaged position, there's photoactivated electron transfer. The Flavin cofactor, that's a fully reduced FADH molecule, absorbs a photon to form an excited state. I've written here as FADH minus with a little star, it indicates it's excited. And then the subsequent electron transfer from the excited cofactor generates a non-canonical FADH CPD radical pair. Now in step 3, shown here, catalytic bond breaking of the lesion occurs. The excited electron induces bond breaking event, and then there's electronic back transfer back to the Flavin adenine dinucleotide to restore its fully reduced state. And finally, the electron hole recombination occurs after the lesion is repaired, and the photolyase releases the DNA and returns to its resting state for the next catalytic cycle as it goes on to repair another damaged piece of DNA somewhere else. So if we want to look at that process in a little more detail, we can consider, for example, a thymine-thymine lesion. That is a pair of thymine genetic bases. If you look at the far right side of this diagram, you can see, in fact, the DNA double helix with a pair of T bases. And normal thymine bases are shown on the right-hand side of the diagram. But if a UV photon comes in, shown as a little lightning bolt, then a damage can occur in the form of a cyclobutene pyrimidine dimer. And so now looking at the left-hand side, we can see in the middle of the diagram, there is a little square between the 5s and the 6s. For those of you familiar with organic chemistry, you'll recognize that square box as a cyclobutane molecule that is now formed between the 2 pyrimidine bases, the thymine bases, and that forms now a dimer, simply a pair of molecules bound together. Now this is bad. If you look at the DNA strand on the left-hand side, you can see that the 2 thymines no longer form rungs of the ladder as they should. They're stuck together on one side of the ladder, so to speak. And you can see the shape of the DNA helix has also been distorted. Now, luckily, if the photolyase comes along with its FADH cofactor in the fully reduced state, another photon of blue light can be absorbed by the FADH cofactor, and now an electron can be produced from its excited state and travel down to the damaged thymine dimer, leaving an FADH radical behind. As the electron enters the thymine dimer, the cyclobutene pyrimidine dimer, it can break up the first bond that's formed, the top bond, breaking up the second bond that's formed, the bottom bond at the 6-6 position, and now is ultimately reabsorbed by the FADH, which discovers its fully reduced state, FADH minus. Now, this process is complex enough, but in some sense, this is only the tip of the iceberg for the whole catalytic mechanism of this enzyme, because that FADH that is crucial to produce the electron, that fully reduced FADH minus state, doesn't come for free. It actually needs to be prepared. And in fact, that kind of fully reduced flavin is even somewhat difficult to handle in the laboratory because it is very quickly reacts with oxygen in the presence of any oxygen to be reoxidized to its usual resting oxidized flavin state. So if we look at the diagram I've shown here, we have a typical example of the photocycle of a flavin. That's a flavin adenine dinucleotide. So blue light comes in and the oxidized flavin, FAD-OX, absorbs a blue light photon. It enters an excited state, and if an electron is transferred to it, in fact a proton as well, it forms a FADH radical. That radical can absorb another photon of blue, green, or yellow light, enter an excited state, the FADH star state, and then capturing another electron in its excited state, form the fully reduced FADH minus state. Now, as I mentioned, that state easily reacts with oxygen and any hydrogen in the vicinity. And as a result, easily returns to its oxidized form, producing hydrogen peroxide in the process. But the enzyme has an architecture that protects that state. And so if we want to understand this process a little bit better, wheels within wheels, we can look just at that single step where the blue, green, or yellow light is absorbed by the FADH radical and the FADH minus fully reduced state is formed. And we see that this has its own cycle within a cycle. If you look to the bottom of this diagram, you can see the FADH radical indicated with the tryptophan chain. That is the hallmark electron transfer chain of cryptochrome and Photolyase systems. I will circle that state in red, and when it absorbs blue, green, or yellow photon, it can be activated to the FADH radical excited state. And now that state quickly reacts in electron transfer reactions and electron transfer cascade with the tryptophan in the so-called tryptophan chain in the enzyme. And it can basically undergo an electron transfer cascade to produce the FADH minus fully reduced state, which is now the, shall we say, locked and loaded form of FAD in the enzyme, which has the electron ready to fire into the DNA to repair a DNA lesion. So suffice it to say that Photolyase photoactivation is a complex multi-step charge transfer process coupled to a very heterogeneous environment of the solvated protein. It is, in fact, on the limits of processes that can be characterized by classical charge transfer theory. And I would say, as a physicist, as a quantum physicist, this is very interesting to me because it tells me the process is at one of the frontiers of quantum physics, a certain quantum classical frontier where these mesoscale physics of charge transfer in this complex environment can produce very interesting quantum mechanical effects to study right at that quantum mechanical classical limit that I talked about in some length in the first lecture of this series. So the environment is very important for this system because if we consider the first stage, the very first stage in the Flavin reduction process where oxidized flavin absorbs that first electron. In fact, recent theoretical studies have demonstrated that the solvent itself is a driving force for downhill hole transfer along the tryptophan chain and contributes to the stabilization of the flavin-tryptophan radical pair. And this is particularly interesting because unlike prevailing notions of this warm, wet, and noisy biological environment where it's assumed that any solvent activity washes out quantum mechanical effects. In fact, there's evidence to show that the presence of the solvent actually stabilizes the quantum mechanical FAD tryptophan radical pair. So this is a very important result, and it tells us a lot about the nature of the enzyme and its ability to stabilize certain important quantum mechanical effects, such as the radical pair that's necessary in that early stage of flavin reduction. So if we take a look at another photolyase system, looking at the left-hand side of the diagram, we see class 2 photolyase in complex with the CPD lesion. It shows graphically a 300 to 500 nanometer wavelength photon. That's that green, blue, or yellow colored photon coming in to activate the FAD cofactor. Now, if we look a little closer into the heart of the enzyme on the right-hand side of this panel, we can see the flavin, the FAD cofactor in yellow, red, and blue. And it has two parts, lumiflavin, that's the tricyclic isoalloxazine ring, shown kind of at the bottom of the figure, the bottom left, and the adenine half of the cofactor that mediates transfer to the CPD lesion, which is labeled there in kind of pink, blue, and cyan kind of color. And now the red dashed lines indicate, as I mentioned before, non-covalent bonds in the system. And we also see some spheres marked with a W. Those are water molecules. And so this shows the importance of non-covalent bonding and solvent effects in mediating this whole process. So it's actually quite a complex process. The figures that I've shown you already don't yet do full justice to the 10 elementary steps, and in fact 7 electron transfer reactions that need to be controlled in order to carry out the 2 crucial bond breaking processes through what is now becoming understood as a universal bifurcating electron transfer mechanism. And so even though there's many different types of photolyase, many different species of photolyase found throughout the kingdom of life, it's now becoming understood that they all follow basically the same essential overall mechanism in carrying out these repair processes. So we could take a closer look at that. We have quantum control of electron transfer in these systems. As I have described so far, the first step is the reduction of the oxidized state of FAD-ox, to its fully reduced protonated state. I am showing here again the usual FAD photocycle from its fully oxidized state on the left-hand side to its fully reduced state on the right-hand side, and the pathways that connect those two. Now, once the fully reduced FADH minus state is formed, it has to absorb yet another, in fact, a third photon in order to produce the excited FADH minus star state, that excitonic state. Now, with that photoactive state, DNA repair can occur by what is becoming understood to be a 1 or 2 step transfer process, either tunneling directly to the CPD lesion, assisted by the adenine part of the FAD cofactor, or in a 2-step process where the electron hops or tunnels to the adenine cofactor and then hops or tunnels directly to the CPD lesion from there. And now we can take a close look at the actual highly complex system of reactions that must be controlled here. In this case, we have the lumiflavin part, the tricyclic part, labeled LFH, and you can see the adenine part in the middle in yellow. And so after photoactivation, there's 2 different forward electron transfer steps that can occur, labeled FET1, FET2, and FET3. And if you now just take a step back and look at this process. It requires forward electron transfer to the DNA lesion on the right-hand side, clockwise around the circle, and back elect transfer, continuing that clockwise motion. Ideally, the catalytic cycle follows only the outer circumference of the circle. Ideally, we have a single tunneling mediated electron transfer step to the DNA lesion. Then we have two bond breaking processes labeled sp1 and sp2 before the final electron return to the isoaloxazine ring, the lumeflavin part. But you see a number of pathways in the middle. There's the less ideal two-step transfer process, FET1 and FET3, but there's also some unproductive back electron transfer processes. So if the electron hops to adenine, it can hop back through BET1A, or it can hop back to the deactivated state, BTT1B, and it's even possible that the electron can get all the way to the DNA lesion, but not fully complete the DNA repair before returning to the Flavin cofactor through back electron transfer process 2. So the enzyme has to control not just the ideal forward and reverse electron transfer processes, but needs to control or even suppress unproductive back electron transfer processes in order to give the system the very high efficiency that it has. Now, this particular diagram is drawn from experiments that were done using the methanosarcina myzete photocycle of this methogen-producing microorganism, but we can look at the photocycle of Photolyase from a more familiar species. Here we have the common fruit fly. And if we look at this photocycle, the time scales and the processes involved are very similar. In fact, basically all the same processes, forward electron transfer processes and reverse electron transfer processes, must be controlled in this Photolyase enzyme, but existing in a different organism, slightly different cellular environment. So this is very interesting because each enzyme is tuned ever so slightly for the organism in which it resides. Now, there's one more item I'd like to draw your attention to on this slide before we move on. If you take a look in the middle of the diagram, I will circle in red the FADH-CPD radical pair. After electron transfer occurs from the flavin to the cyclobutene pyrimidine dimer, we have a radical pair, the FADH radical and the CPD radical, indicated with the little dots, the little bullet characters. And this is understood to be an entangled quantum pair of electrons. How do we know that? Well, experiments have been performed. And so I show here again a similar photocycle drawn from experiments by Zhuang and co-workers, where they showed that the FADH-CPD radical pair remains spin entangled despite the spatial separation of the electrons. And it undergoes singlet-triplet dynamics that govern bond breaking and recombination pathways in the DNA photolyase complex. This experimental evidence comes from studies that they did using Photolyase from the E. coli bacteria, and the way they were able to demonstrate this was by showing that the DNA lesion, the dimer repair, was magnetic field dependent. They studied this in mutant photolyases as well as an engineered cryptochrome molecule in order to replicate these effects across a variety of different systems, validating the radical pair mechanism framework that is used to characterize these kinds of magnetic field sensitive reactions. So in the conventional radical pair model, there is a pair of electrons. Electrons usually are paired up in a molecular bond, but when the photoexcitation occurs, now one of the usually static electrons in the pair becomes excited, and as we've discussed, of course, is transferred to the DNA lesion. Now, this can undergo singlet triplet interactions, dipole-dipole coupling to a third spin in the system. Every electron or some nuclei have spin. Will sensitize the quantum mechanical singlet and triplet states so that singlet triplet dynamics are sensitive to the presence of a weak external magnetic field. And this kind of mechanism is called a radical pair mechanism that can change the electron transfer reaction yields based on the presence of the magnetic field. So weak external magnetic fields, if you hold a magnet up to the system with a varying magnetic field amplitude, that's the field strength, or the magnetic field inclination, that's the angle the field is pointed at, will modulate singlet triplet interconversion rates via Zeeman splitting and or hyperfine interactions with the nuclei, and the result is that the DNA repair yield, the percent success rate of the DNA repair process can be controlled to some degree simply by putting a magnetic field in the presence of your Photolyase repair system. So Peter Hoare from the University of Oxford has commented that remarkably, the rate at which the repair process proceeds depends on the intensity and direction of the external magnetic field provided, and this is a hallmark of the quantum mechanical radical pair mechanism. Now, the activity of the enzyme can also be influenced by other aspects of the protein environment. So, for example, time-resolved absorption spectroscopy experiments have revealed that the Flavin adenine dinucleotide can toggle between its fully reduced FADH- state and its semiquinone state during the recombination after FAD reduction. These transitions are governed by proton-coupled electron transfer with two distinct mechanisms that can switch near a pH of about 6.5. Now, this is very interesting because changes in pH influence proton-coupled electron transfer pathways, the recombination quantum yields, and the FAD spectra, which is analogous to other pH-dependent color shifts that have been demonstrated in firefly bioluminescence. And this once again reveals the Photolyase enzyme's finely tuned catalytic sensitivity. Now, the Flavin cofactor itself is in some sense an ideal photosensor because of these interesting and unique property it has. So the Flavin adenine dinucleotide adopts a U-shaped or hairpin kind of conformation, as you can now see here, that positions the isoalloxazine ring for efficient photoinduced electron transfer in Photolyase and in cryptochrome, for that matter. So unlike two electron characters such as NAD, that's nicotinamide adenine dinucleotide, FAD, Flavin adenine dinucleotide, stables the semiquinone radical intermediate, enabling magnetosensitive single electron transfers that are essential for the operation of the radical pair mechanism that I described. Now, in addition to the Flavin chromophore, of course, I mentioned there are other accessory chromophores, other antenna cofactors, such as the diazoflavin 8-HDF, or a variety of different pterins and or folates, such as MTHF, that broaden the absorption spectra, ultimately improve the optical absorption cross section of the enzyme, and funnel excitation energy to FAD, enhancing light harvesting efficiency in the enzyme. And of course, we can't forget these tryptophan chains. In fact, there's a whole network of tryptophan in Photolyase, which I've shown here. You can see surrounding that U-shaped hairpin FAD, conserved tryptophan triad of 3 or even a tetrad of 4 tryptophan residues, quenches the initially excited FAD state in a stepwise electron delivery mechanism to form a redox active FAD radical minus, tryptophan radical plus, radical pair. And that fourth tryptophan, where it's present in certain species of photolyase, extend the tryptophan chain to the protein surface to make it easy for the enzyme to capture extra electron from the solvent environment. So this surface exposed tryptophan can be reduced by solvent molecules to replenish the electron that it donates to the flavin. It also has been hypothesized to engage in downstream signaling in cryptochrome. And this is very interesting because it could couple FAD photochemistry to broader cellular responses, for example, in some cryptochrome dash species, which has also been shown to demonstrate photolyase-like DNA repair. And so also, finally, flavin tryptophan photooxidation can trigger functional and structural changes in the enzyme. So the protein architecture and the solvent network stabilize the radical pair, shaping its lifetime, anisotropy, and even its recombination pathways in a highly intricate kind of quantum control mechanism. Now, tryptophan is particularly interesting because it can act as a photoreceptor itself. So in the ultraviolet resistance locus 8, the UVR8 photoreceptor, this is a plant photoreceptor that in fact has a tryptophan network as its sole light harvesting antenna system. There's no additional prosthetic chromophore like a flavin or a pterin or a folate required in this system. It achieves quantum efficient UV sensing, funneling excitation energy through the collective tryptophan states themselves. So this is really fascinating, and it could have a great deal of importance for biomedicine because these tryptophan chains couple protein structure to environmental signals, including light signals, in diverse photosensitive systems, including in my own work. We've studied cytoskeletal proteins and amyloid fibrils associated with various states of cellular health and disease. Dysregulated tryptophan metabolism is implicated in cancer progression and in some neurodegenerative diseases. And so deepening our understanding of the light active properties of amino acids like tryptophan is likely to have significant implications for biomedicine and biotechnology going ahead. For example, in my own work, I've studied the presence of superradiance in tryptophan networks in the cytoskeleton, the structural skeleton of the cell. So this made the cover of the Journal of Physical Chemistry B. You can check it out. We demonstrated experimentally entanglement-enhanced collective emission that accelerated photon release, and this could be important for potentially dissipating harmful ultraviolet excitations in the cell before they damage other cellular components. Now, the presence of superradiance is accompanied by another effect called subradiance. Rather than rapidly re-emitting captured photons, a subradiance effect does the opposite. It stores the energy from a photon within the tryptophan array, preserving the exciton for subsequent photochemical or signaling events. Now, what is really interesting is that either or superradiance or subradiance could play a significant role in photoprotection. For example, microtubules or actin filaments in the cytoskeleton, the structural network of the cell, could employ tryptophan superradiance to dissipate potentially harmful energy, or subradiance to capture and ultimately dispose of energy in the form of UV photons to safeguard the cytoskeleton and other cellular components, for example, inside the nuclei, from photo damage. Now, we've looked into this more deeply in a recent paper published in Frontiers in Physics with colleagues of mine. And in fact, the whole idea of superradiance and subradiance in the cell suggests the possibility that collective coherent phenomena may also enable other key cellular functions beyond photoprotection and could even be involved in cellular communication, redox regulation, metabolism, etc. And so this really raises the question of whether the cell is performing certain rudimentary forms of quantum information processing in the system. So, let's recap. There are a number of optimization challenges that need to be met in the cell for a DNA repair enzyme like photolyase. First of all, repair enzymes must rapidly locate rare UV lesions within the crowded cellular milieu, and a vast combinatory search optimization task needs to be completed, which is both constrained by the limits of diffusion and macromolecular crowding. So really, at this point, the question is whether these classical searches are additionally enhanced by quantum mechanical effects. Now, looking into the noise resilience of the system, in fact, a very high catalytic efficiency is maintained amidst thermal fluctuations that can lead to random charge dissociation, premature recombination, electron loss, incomplete lesion repair, and or collateral damage by the excited electron. So the fact that the enzyme is able to carry out all these steps with high efficiency, low losses, and basically non-existent collateral damage is an incredible testament to the noise resistance of this quantum mechanical mechanism amidst the thermal environment. Now, finally, a third, we have the importance of the synergy of the process. Photolyase coordinates a series of Flavin photoactivation reactions. As oxidized FeD is excited to produce an FeD radical, which is protonated and excited again. Now, the final fully reduced excited FeDH minus star state then leads to forward electron transfer to the DNA lesion, controlled bond breaking, and electron recovery. And all these steps need to happen, positively and constructively once again in this noisy system. So this is a real optimization challenge, and to me, it's a testament, to the advanced architecture and ultimately fundamental design principles that go into the coordinated action of this enzyme. Now, we are learning more about these systems all the time through ultrafast dynamics and imaging using a variety of experimental methods. So for one, X-ray crystallography captures atomic resolution snapshots of radical pair stabilization and induced conformational changes within the photolyase active site. Time-resolved spectra on the femtosecond and picosecond scales from transient absorption spectroscopy are used to map FAD redox switching, tracking the semiquinone formation, radical pair dynamics, and back electron transfer, which I've showed you on a number of slides. All these timescales have been mapped out through spectroscopy. And increasingly, the structural dynamics of the system, the actual ultra-fast dynamics of the enzyme as it carries out these catalytic processes are being characterized using, for example, recently, X-ray free electron lasers, which allow basically a crystallographic movie of the system to be recorded. And this is being enhanced with additional slower millisecond observations by time-resolved cryo-electron microscopy. And I've shown just an exemplary picture from the European X-ray free electron laser on the left-hand side of the slide. Now, all of this data needs to be interpreted, of course, using ab initio, multi-scale quantum classical simulations, quantum chemical simulations to model chemical bond cleavage, electron and proton transfer pathways, and solvent conformational transitions in order to fully understand the data that we're even looking at when we use these advanced experimental techniques. So all of this has important implications for biomedicine and biotechnology. For example, designer photolyase-like DNA repair enzymes or even photolyase extracts have been proposed for use to protect tissues from UV radiation, preventing the development of skin cancer, as well as reducing the risk of tissue photoaging. Study of an enzyme like photolyase is important for developing our understanding of the enzyme for enzyme engineering and the design of high-yield photocatalysis based on the FAD tryptophan architecture found there may help to address and ultimately treat metabolic diseases linked to defective redox enzymes. Now, this is all very exciting because the possibilities for quantum biotechnology based on discoveries made mapping DNA repair dynamics by femtosecond spectroscopy and ultrafast X-ray crystallography really presents an important opportunity to understand the synergy of the elementary chemical steps that are involved in this reaction. And so the photolyase enzyme is ultimately an important experimental prototype for studying collective synchronization of quantum effects in enzymatic systems and ultimately reproducing them in designer de novo enzymes as quantum biotechnology becomes more prevalent in our society. In conclusion, photolyase exemplifies quantum biological control. Electron tunneling, radical pair spin dynamics, and tryptophan relay networks synergize together to achieve optimal DNA repair quantum yields simply under ambient light in the warm, wet, noisy cellular milieu. A unified framework has been developed to understand the mechanism of these systems and to explain the nuances of the electron injection into a DNA lesion. Now, this unified scheme is proposed to involve a bifurcated process involving either one-step or two-step tunneling and or hopping. And similar architectures across photolyase enzymes support a universal electron transfer bifurcation mechanism. And now this has enormous promise for technology translation because the convergence of ultrafast biophysics, spin chemistry, and high-resolution structural biology, even time-dependent, time-resolved structural biology, is likely to drive breakthroughs in the development of DNA protective drugs, engineered photocatalysts, and other forms of quantum biotechnology, such as biosensors and other designer redox enzymes. So if you've enjoyed today's lecture, please download your copy of Physical Principles of Quantum Biology. It's your guide to quantum biology. You can find it on the archive at this link. It's available now for researchers, students, and curious minds ready to venture beyond classical biological explanations. And if you have questions, don't hesitate to contact me, Dr. B, at nbabcock@gmail.com. And please continue to check out these lectures on my YouTube channel. It's been a pleasure talking to you today, and I look forward to speaking with you again next time. Have a great day.